Okay. Alright, okay. So let's do the genetics way, okay? Sorry. Over here, most of you say that, okay. Yes, most of you think that, okay, since all the puppies were black, right, this female dog must have only given the dominant black allele, or her gametes had a big V, right, and therefore all the offspring were black. That seems possible. But our discerning friend over there just pointed out a counter example. He said, well, we only had 3 puppies. What if it's by chance? Okay, so sometimes you do have to take note, right? If your sample size is very, very small, it may not be statistically significant. So the very, very last one, genetics, we will deal about chi-square test to test for significance, okay? But if the sample size was big and all the offspring were black, well, you probably can conclude that it's true that she was a pure breeding black dog, all right? Try question 2. You want to copy this? Logic, okay, do it quick, okay, if you want to copy anything. If you copy any slower, you can go to the recording and slowly copy, okay? Alright, if you can do it fast, then copy in point forms. Alright? So by genetics, it seems correct, but we also have to take note of sample sizes when we do such things. Which leads us back to last week, last lecture, actually, when we talked about Mendel. Mendel used pea plants, which gave him a lot of progeny. So therefore, his numbers were quite statistically significant. When he came to a 3:1 ratio, he found it was a typical law. When you cross 2 heterozygotes together, you can get this sort of thing. And it was significant because his numbers were very large for the offspring. Okay? Ready? Going down already, alright? Those of you who finished copying, start to read to question 2 then. Okay? Try and work out question 2. Alright? So in question 2, you have poppies. Poppies may have plain petals or petals with a large black spots. So there are 2 possible phenotypes. Either you are plain or you have spots on your petals. Now it says that if 2 plants with spotted petals are crossed, the offsprings always have spotted petals. What can you conclude from this sentence? If 2 plants with spotted petals are crossed, the offsprings are always spotted. They have the same phenotype as the parent. What can you conclude from this? They must be? Must be what? Yes, Bridget? They could be recessive. They could also be dominant. We can conclude that they are pure breeding. Right? Possible? Right? Okay? Now, a cross between unspotted and spotted plants sometimes produce offsprings which all have spotted and sometimes half. Okay? So, first thing over here, right, this sentence tells us that they are homozygous, they are pure breeding. Okay? Because the offsprings always look like the parents and phenotypes. Now, when you say they're pure breeding, the homozygous, they could either be homozygous recessive or homozygous dominant. We don't know yet. We need the next sentence to help us figure that out. Okay? Right? So now the next sentence says, if you cross unspotted with spotted, you produce offsprings which all have unspotted petals sometimes, and sometimes produces. So we realize that spotted plants cannot be homozygous dominant, because if they were, right, all the offsprings will be spotted, and therefore unspotted is dominant. Okay? Alright? So when you cross unspotted with spotted, all the offsprings have unspotted, which tells us unspotted must be the dominant one, not spotted. Alright? Okay? So, it says sometimes when we cross this way, you can get half spotted, half unspotted. So if spotted is homozygous recessive and unspotted is dominant, then this half-half is a 1-to-1 ratio. Right? So it's your typical Mendelian test cross ratio. Alright? Okay. So this is how you can work out the genotypes based on this description or the phenotypes of the offspring. Okay, this is a very simple monohybrid question. Okay, all right. So you don't have to copy this, right? This is, for example, if you had thought spotted was dominant. Okay, first sentence tells us we know that, okay, they must be homozygous, right? Spotted is homozygous. And if you chose to say that spotted was dominant, then you'll find that when you cross spotted with unspotted, all of them will be spotted, which was not what we saw. Correct? So this is wrong, right? So what we really saw was that unspotted was dominant, because when you cross unspotted with spotted, offsprings were all unspotted in some cases. That means unspotted was a dominant case. Alright? Okay, so if you were to show this in a genetic diagram, which is a simple monohybrid diagram, you don't have to copy this, right? If you understand this, right? Your unspotted cross spotted or unspotted. So in this case, that particular parent was homozygous for unspotted. Okay? Then you cross it with a spotted. Spotted must be recessive. Okay? Where else, in some other cases, when you cross spotted with unspotted, those unspotted are the heterozygous ones. They show the dominant unspotted phenotype, but they're heterozygous. You cross it with the spotted, which is recessive, you get a 1:1 ratio. So this is your typical Mendelian test cross ratio, right? Okay? And this can tell you the identity of the unspotted parent that you use to cross. Okay? So this is basically practice 2 questions to sum up what we've learned so far under monohybrid inheritance. We've talked about test cross in our last one, and this is an example how you will work it out. Okay, if you're asked to do a test cross to solve a problem. Okay? If you figured out that spotted was homozygous recessive, you're actually doing a test cross in this case. Alright? Okay? So far, right? Okay, let's go on. Alright, so let's do some genetic tips, okay, to round up our monohybrid inheritance. If you turn with me to page 13 first, okay, let's finish up what we did on the last lecture. Okay? These are some of the typical Mendelian monohybrid ratios which you should be familiar with. Okay? If you get a 3:1 phenotypic ratio in the offspring phenotype, you can be pretty sure that the parents that will cross to produce these offsprings, their genotype would be, they will be, yes, you agree? If they a 3:1 ratio. It's a typical Mendelian ratio when you cross what with a what? Heterozygous cross heterozygous, right? Okay? Right, so big A small a cross big A small a will give you 3:1 ratio of dominant to recessive phenotypes. Okay, this should be quite straightforward to you. If you cannot immediately come out of this, you need to revise a little bit your O-level stuff. Okay? Alright? Okay? So the ratio is a 3:1 ratio. We cannot see page 7 again. Alright? So the 3:1 ratio is actually a phenotypic ratio. But actually, if you look at the genotype, right, the ratio is actually 1:1. If you look at the genotype, 1 would be homozygous dominant, showing the dominant phenotype. 2 out of 4 will be heterozygous for the offspring, and 1 out of 4 will be homozygous recessive. Okay? Right? So remember, the 3:1 ratio is a phenotypic ratio. Alright? Okay, so these are good genetic tips for you to remember as you apply them. Alright? Okay. Now, the 1:1 is a typical Mendelian test cross ratio. This ratio is obtained when you cross what with a homozygous recessive individual? Answer will be? Friend there? Yes? With the orange bottle cap. Quick answer? Renner? If I cross the homozygous with homozygous, will I get a 1:1 ratio? Huh? So if I cross the homozygous dominant with homozygous recessive, will I get a heterozygous, right? So when you cross a heterozygote with a homozygous recessive individual, you get a 1:1. Now it's okay if you cannot remember right now. You probably want to practice drawing the genetic diagrams a few times to prove to yourself every time you cross a heterozygote with a homozygous recessive, you get a 1:1. Once you've drawn it maybe 3 times and you proved it to yourself, it's in your brain. So it's better than trying to memorize it. You cannot immediately memorize what 1:1, 3:1 is I recommend go back to basics, draw a simple genetic diagram. Draw it a few times until you're very, very sure, oh yeah, it's true, it's like that one. Okay? Okay? Alright? So your genotypic ratio is actually 1 heterozygotes is to 1 homozygous recessive, the 1 to 1, alright? Okay, now I'm going to go on to what we call dihybrid inheritance. This one I need to pay attention to, okay? It's different from monohybrid inheritance, especially when you draw diagrams. For the happy VCA children here, or people here, I'm sure you're very happy. Just listen to some of the things, alright? Now, dihybrid inheritance is the inheritance of two pairs of contrasting characteristics simultaneously. So, example, you have a seed. This seed has a shape and has a color. You're trying to see if I cross these two plants, maybe this seed is round and yellow. This plant gives you seeds which are round and yellow, and this plant gives you seeds which are green and wrinkled. So green is the color, wrinkled is a characteristic of the shape, right? Now you're trying to see if you do the cross between these two plants, what is the outcome of the seeds? You're trying to look at two characteristics simultaneously. Okay? So Mendel did this actually, right? He wanted to say, hey, if I do a round yellow cross green wrinkled, what happens to the progeny? Is there any relationship, right? Would green always go with wrinkled? Will round always go with yellow? Or is it the inheritance of these two characteristics independent of one another? Okay, so the question we want to ask ourselves is the inheritance of two different characteristics, are they related to one another? Or are they independent of one another? Okay? So let's take a look at this. Our big R, we should assume that round is dominant, small r recessive or wrinkled. Okay? And yellow is dominant, and small y will represent the recessive allele for green. Okay? So, in our genetic diagram, our format is parental phenotype. So let's assume this first cross, okay? Mendel had many pure breeding plants. So he took one happy pure breeding plant with round yellow seeds, and he crossed it with another pure breeding plant with wrinkled green seeds. And what do you think the outcome of his F1 generation phenotype was? It would be, draw meiosis, get our gametes first, right? Okay, so now how do we get gametes in this case? Remember his first law, right? His first law says that each gamete should contain only one allele for each gene. So over here, right, we will expect this, alright? You don't draw separate circles like R is one circle, Y is one circle, no, right? Gametes has the sum total of whatever alleles that should be going down to the offspring, alright? So you just put one big R and one big Y for this. There's only one kind of gamete because it's pure breeding. And this gamete here is pure breeding, will only give you one kind of gamete. So remember, put R and Y together, don't put R one circle, Y in one circle. Doesn't work this way, right? Alright? Okay, just like your gametes, right? You have 23 chromosomes in the gamete. It's not singular, each gamete got one chromosome. Alright, okay? So, when you cross together the F1 genotype, he found that all of them were round and yellow. That proves to him that round was dominant, yellow was dominant. Right? How do you write the genotype for something like this? Right? You put them together. So big R, big Y, small y. Okay? That's how you write the genotype. Okay? So big R, small r is heterozygous, but because of the dominant big R, you are round. Big Y, small y, right, coming from here. This one donates one big Y, this one donates one small y. This one, because of the dominant big Y, you are yellow. Okay? Let's go further from here. Now what happens when you self your F1s together? Now when you self a F1, so he took that plant that was showing the F1, that was showing the round and yellow phenotype. Okay? These are called double heterozygotes. They are heterozygous for this gene loci, and they're heterozygous for this gene loci. Okay, so it's a double heterozygote. Okay, so this double heterozygote, when selved, gives rise to gametes. How do you draw gametes now when your genotype, you're not a pure breeding plant? Okay, same principle, you need to put one allele into the gamete, right? So how do you do it? Let's take a look. This is one way. So you choose big R goes with big Y into one type of gamete. What other combinations? Big Y can go with small y in this combination. Small r can go with big Y, and small r can go with small y. And that's how you write gametes when you're looking at dihybrid inheritance. Remember, okay? One gamete can only take one allele, but you make sure you do them together. Alright? Know how to do this? Okay. Now that you got a combination of 4 times 4, you will not draw your random bars, you will do a Punnett square, alright? Okay, so this is a Punnett square. You put the gametes, the 4 possible gametes that you found, right, here. And this will very easily ensure that you get every possible offspring genotype out. Otherwise, you'll forget things, okay? Now, I'd like you to analyze this chart. This is a very important chart. The more you are familiar with this, the faster, I promise you, you will be when you do genetics MCQ questions. Okay, so this is first time I'm giving it to you. As you work through it, try drawing this yourself. After you draw about 3 times and you particularly know this very well, you can say, okay, I know at this part, this is this. You will be able to do your genetics questions very much more easily because you know what is in the Punnett square for a cross between a double hetero and a double hetero, okay? But now let's go through very quickly. So how do you write the genotype of the F2? Like this. If you look at these crosses over here in this way, I like to put it this way in this format so it looks in this pattern, alright? So if I put my big R, big Y here, this is my big R, big Y, homozygous dominant for both gene loci. In fact, all these 9 will be round and yellow, phenotypically, of which only 1 out of 9 is actually homozygous for both. It's a double homozygous. Okay, the rest are actually varying levels of heterozygosity, right? Okay? Okay, what about this then? Do we get other phenotypes besides round and yellow? 9 out of 16 would show my dominant phenotype of round and yellow. Okay? 3 out of 16 would show round, which is dominant, and for the color, they would show the recessive phenotype. Okay, so 3 out of 16 will show dominant for one phenotype, dominant for one characteristic, and the recessive phenotype for the other characteristic. Then the last 3 here, this corner, would now switch over. You will show the recessive phenotype for that first trait of shape, but you will show the dominant phenotype of the other traits. Okay? And lastly, the guy in the corner here, this is the 1 out of 16 who will be purebred homozygous recessive for both traits. Okay? So you do this enough for double heterozygotes, you will find you always get a 9:3:1 ratio. Please understand what the 9:3:1 means. 9 represent the individuals that show both dominant phenotypes. The other 3 is dominant, then recessive phenotype. The other is the recessive dominant phenotype. And the last 1 out of 16 is the recessive phenotype. Okay? Okay? Okay. So let's look at this, right? When he did the cross at the end of your Punnett square, we can sum it up nicely using this kind of legend. You see over here? 9 out of 16 would show the round and yellow phenotype. So they all definitely will have one big R and one big Y. That's how we can sum it up, correct? For the 3 out of 16 that show the round and green, they all have at least one big R and 2 small Ys in order to show the green recessive trait. Then the other 3 out of 16 will have 2 small Rs and at least one big Y. And the last one, the green and wrinkled, will be small r, small y, small y. Okay? Know this ratio well. It's your typical Mendelian dihybrid ratio when you cross 2 double heterozygotes. So far, okay? We still haven't answered our question, right? But we know how to draw a Punnett square for a dihybrid, and we know how to draw a genetic diagram to show parental F1, F2. So far, yes? Okay, so what's the question we asked just now? Right? Okay, let's talk about it, right? Now we asked whether the inheritance of these 2 traits were independent of each other or are they separate. So let's answer that question now, okay? Now I'd like you to look at page 12 and let's look at each characteristic out of the 9:3:1 phenotype ratio that we saw. If you look at each characteristic individually, right, what do you see? If you look at seed shape, What is the ratio of round is to wrinkles? Quick answer will be, if your math is good, it is 3:1. Alright? Right, using the F2 phenotype ratios, right? From this we see, okay, actually if you look at each characteristic individually, the characteristic for seed shape gives me a 3:1 dominant to recessive phenotype outcome. And if you look at the seed color, disregarding the seed shape one, right, you also get a 3:1 ratio. What can we infer from this observation? Did the inheritance of seed shape affect the inheritance of seed color? Or were they independent of each other? Right, I see some of you, doesn't affect, right? Yes, they were independent of each other, right? Okay. We realize that for each trait, the ratio of the F2 generation exists like a typical 3:1 for each trait. It's a typical 3:1 monohybrid for each trait. Remember my F1 was a double hetero? Right, if you look at each of the traits itself was hetero cross hetero, it gives you a 3:1, 3:1. Okay? Right? So answer the question. It was independent of each other. This is on page 11 if you want to write something to answer the question. Right? Can we explain why? Why is it independent of each other? Why didn't the inheritance of seed color affect the inheritance of seed shape? Okay? The reason is because these two genes are found on different chromosomes. And we learned in meiosis that independent assortment allows for equal proportions of gametes in all kinds of ratios, in all four ratios in equal proportions. Okay, so a lot of independent assortment. So if you take a look, right, these two gene loci on different chromosomes, but independent assortment, big R can assort big Y, equally as likely as big R can assort with small y, equally as likely as small r can assort with big Y, equally as likely as small r can assort with small y. You get all four possible gametes, right, in this dihybrid cross because of independent assortment. Each of these characteristics were on different chromosomes. By the law of independent assortment, yeah, you can equally put them out. All these gametes will come out in equal proportions. So when you draw your Punnett square, right, you realize, yeah, the proportions are going to fill in nicely. No one gamete will dominate compared to others. They'll all be in equal proportions. And therefore, you get the 9:3:1 ratio, which when you look out, each trait was actually acting independently from another. That aside, doesn't mean that therefore you can draw your dihybrid as two different monohybrid crosses. Not allowed, okay? Never draw your dihybrid diagram as two different monohybrids. That is wrong. You have to draw as a dihybrid as I showed you just now. Okay? But knowing this important thing helps you with MCQs when you want to deduce out the identity of a genotype of a particular characteristic from this. Okay? Okay. Ready? Okay. So let's do very quickly. If you didn't understand what we're looking at again, we're looking at round, the gene loci for shape being on this chromosome, and the gene loci for color on another chromosome. So by the law of independent assortment during meiosis 1, right, when you randomly put your homologous chromosomes out, right, what you have is two possibilities, right? Either like this or like this, right? I think you're very familiar with this by now. Okay, so this subsequent thing allows for the different combination of my alleles, big R with big Y, big R with small y, small r with big Y, small r with small y in equal proportions. There is no particular particular gamete that is favored. There's a higher number of them. And therefore, when you draw your nice Punnett square, everybody has an equal nice chance to give you a 9:3:1 ratio. Okay? Right? So let's run through again his two laws. Right? His observations led him to these two laws, which we learned today, right? That a copy of allele for each gene is present in each gamete. That was Mendel's first law. So remember, when you draw your gametes, please put them nicely. One circle should have one allele for each gene locus. And then different combinations of alleles are possible in equal proportions by Mendel's second law, the law of independent assortment. So far, okay? Okay, if this you're okay, then we'll try some practice questions. Alright? Over here, I'm going to give you a minute to do this. Now, what we're going to do right now in this table is I want you to come out with all the possible phenotypes. Sorry, this is the phenotype, round and yellow. I want you to come with all the possible genotypes for all plants with round and yellow. If you can do it now without looking at the Pilot scale on page 11, please do it. If you need some help, page 11 will help you. I'll give you a minute. Write down all the possible genotypes that may be round and yellow, all the possible genotypes that are round and green, and those which are yellow and wrinkled. So these three characteristics all have at least one dominant characteristic. Okay? Give you 1 minute to do this. To page 11 if you need some help. Or you can work it out. If you can work it out, do this. Okay? Time's up. 1 minute over, right? Let's check. These 4 possibilities will all give you round and yellow. Right? So if you get a pea plant with a round and yellow seed, you can't be very sure which genotype it is, okay? Alright? It could be homozygous dominant, it could be hetero, double hetero, or a combination of others. If you're round and green, these are your 2 possibilities. Being green is easy. Being green is recessive, so it's small y, small y. But if you were round, you could either be homozygous round or hetero round. If you were wrinkled yellow, wrinkled is easy, wrinkled is recessive, so you're small r. But if you were yellow, well, you could either be big Y, or hetero, big Y, small y. So far correct? Okay? So now on page 14, I'm going to talk to you something about something called the dihybrid test cross. We've dealt with the monohybrid test cross before, right? So I hope you're quite familiar. A test cross is for you to find out the identity, hopefully, the genotype of this individual who displays a dominant phenotype. Okay? Alright? So let's take a look. Now, I'd like you now to try and practice this for yourself. I've shown you one time how to write the gametes. Now, if I give you now this round yellow parent, which is a double hetero, and I ask you to do a test cross within a wrinkled green fella, can you show me the 4 types of gametes, just the types of gametes that being found for this round yellow double hetero, and the types of gamete that being found for the wrinkled green? If you need any help at this point, you're not too sure to do a gamete, raise your hands, your tutors are here, we will help you. Okay? Very quickly, I'd like you to practice drawing this genetic diagram, draw the gametes out. Raise your hand if you need help, we will help you. Pencil or pen, up to you. You know how to do this? Okay, anybody need help? Okay, so remember the law, Mendel's first law, okay? In every gamete, there can only be one allele, either big R or small r, big Y or small y, cannot be two R's or two Y's inside there, okay? In every gamete, there can only be one, okay? Check, okay? So this double heterozygote, so the first thing when you're going to give gametes is, please draw an arrow, write the word meiosis. There, okay? We give you marks for this. Tell us how your gametes arose magically on your paper, okay? Draw an arrow, write meiosis. Check, is this your gametes? Yes? There can only be one allele in one circle, okay? Now, here, are there four types? Are there four gametes? Yes. You only have to write the types. Don't have to write 4 times. If you're homozygous recessive, you only have one type. So we only write the types, okay? From this onwards, let's take a look. Give me your ratios. What is your typical expected dihybrid test cross ratio? Write your ratio yourself. Okay. Now, when you write the F1 genotype, notice we cluster the common alleles together. So, for example, for wrinkled yellow, I write it as small r, big Y, small y. Yes, one small r came from this guy, one small r came from this guy, one big Y came from this guy, and one small y came from this guy. So can I just write it this way then? Small r, big Y, because it came from this guy. And then small r, small y came from here. So I write it like that. Is this correct? No. Okay, do take note. It's a common mistake sometimes. Remember, we always cluster it together. So it should be small r, big Y, small y. Okay, and not written like this. Okay? Okay? Alright, so if you're looking, right, remember we looked at the law of independent assortment, right? Independent assortment means the equal proportion of all these gametes can be found because these 2 gene loci are on different chromosomes. Okay, so all these 4 gametes are going to occur in equal probability. Okay, alright? And because they occur in equal probability, when you test cross it with a homozygous recessive individual who can only give one kind of gamete, you get a typical Mendelian dihybrid test cross ratio of 1:1:1:1 every time you cross a double heterozygote with a double homozygous recessive individual. This is your typical Mendelian dihybrid test cross ratio. So anytime when you see a genetic diagram and they give you 1:1:1:1, you're very happy already. Okay? One individual must be double homozygous recessive, the other individual must be double heterozygote. Alright? Okay? Okay. All the other crosses, there are many. You can work them out. You don't have to worry about them. Okay? But if you got time, you got the free time, go and work them out for fun. Okay? But we just tried the other guy. The other guy who is showing both the dominant phenotype of brown and yellow, but it's not a double hetero. He is homozygous dominant. Big R, small y, big Y, big Y. What do you think is the outcome of the cross when I cross a double homozygous dominant with a homozygous recessive? Or the phenotype will show the dominant phenotype. Okay? Alright? Okay? Okay, so if you are quite happy with this, then I'd like you to go home to try on page 15. That's practice question 3. If you're working on a piece of paper, try drawing your diagrams, get familiar with how to draw gametes, how to work out answers. Okay? These answers, I'll put it on the VJC Google site at the end of next week. So I won't do practice question 3 here. It's for you to work out a piece of paper, slowly practice drawing. The more you practice, the more comfortable you are doing 100 squares, the better you will be. Okay? But I will do question 4 with you. Alright? Now if you look at question 4, we are going to take a look at plants that are, okay, so this variety A of flax is resistant to the rust fungus disease caused by race 22 of Melamsora linii. but it's susceptible to raise 24. Okay? What else? There's another variety B, which is susceptible to raise 22, but resistance to raise 24. The F1 hybrid of two varieties, when you cross A and B together, is resistant to both races of fungus. Okay? So let's take a look at clues at this. Well, some clues you can pick up from this to help you interpret. Your task today is to find out what is the genotype of variety A and what is the genotype of variety B based on the question here. What clues can help you? This is the first clue, right? When you cross A with variety B, you only get one phenotypic outcome in your F1. This is one interesting thing, right? In what situations will you only get one kind of outcome when you cross two together? Homozygous, I hear someone say, right? Some idea the parents somehow must be pure breeding in some way, right? That's why there's only one kind, right? Because each parent can only give one kind of gamete. Then you realize, you look at F1 itself, what phenotype is it displaying? Right? It's showing resistance to both. Okay, so does it tell you whether being susceptible or being resistant is the dominant trait? Okay, right? After you've done this, they give you on page 16 the results of the cross from the F2 generation. These numbers are very useful to you. Chemists will never tell you the ratio, they will give you the numbers. You will have to calculate the ratio. If you look quickly, what kind of ratio do you think it falls into? 9331. And this 9331 F2 ratio for phenotypes, what must the genotype of the F1 be? 9331 was a typical Mendelian dihybrid test cross ratio, right? So the parents must be double heteros, right? So now we know the F1s are double heteros and the parents are pure breeding. Can you try and work out that? I think cover my answer. I was supposed to cover the answer. Fill the blank first. I give 1 minute to fill the blank on page 16. Fill in the 2 little blanks there. Then I'll show you the answer. Go. Okay, I already wrote everything there already. Just left 2 little blanks, okay? Fill those 2 little blanks under the answers. Ready? Set. Check. Correct? Haven't do yet? Right, Nina? Those 2 little blanks. Done? Not, yes? Okay, go. Read through that little paragraph and fill that 2 little blanks. And then work out for me the genotype of A and B. Okay? I give you 2 minutes to read through and do that. I don't have much time. Do hurry. One more minute. Work out the genotype of the parents. Okay. Alright, some of you got it already, alright? So Moving on, okay? Can? These two lines you filled in already? Can? Yeah? Answer? Ready to show answer for parents? We figured out that F1 must be a double hetero. The parents A and B must be pure breeding. Now the figuring out of what kind of pure breeding. Pure breeding just means homozygous, right? Does not mean anything, right? So this is A. Yes, A is big A, small b. This is pure breeding. And variety B is small a, big B. Alright? Okay, what's going on? So last genetic tip for this set of lecture notes. Let's talk about the 9:3:1 ratio. That is a typical phenotypic ratio when you cross two double heterozygotes together. And a 1:1:1:1 dihybrid test cross ratio is when you cross one double heterozygote with a homozygous recessive individual. And why can you get such nice phenotypic ratios for Mendel when you do such dihybrid crosses? Simply because there is independent assortment of chromosomes into different gametes. When you look at this dihybrid inheritance, you're looking at two different traits carried by two different genes, and these two genes are on separate chromosomes. So by the law of independent assortment, you can equally assort them out into different gametes. Okay, all equal proportions of gametes are possible. I stress this a little bit here, it sounds so obvious, because later you'll go into non-Mendelian genetics, which the H1 do not need to know, but H2 yes. And then you realize that it's not going to apply for those other cases in non-Mendelian genetics. Okay? So now I'm just going to stress for typical Mendelian dihybrid ratios, we are looking at two different genes on two separate chromosomes. The law of independent assortment applies. Okay? Ready now? Please turn with me to part 2 of non-Mendelian genetics, lecture notes. For the H1, you can still hang around. Okay? Now I'm going to talk about what are the situations where you don't see Mendel's ratios applying, okay? Why do these situations, why do his laws not cover all these situations? Because number one, not all organisms are diploid, so you can't have the law of segregation, right? A gene may have more than 2 alleles. So far Mendel was very lucky. For his pea plant, all these characteristics was 1 gene, 2 alleles, right? So he was very lucky. But you will come across situations where 1 gene may have more than 1 allele, and it affects the kind of outcome you see later. It may not always get Mendel's ratios. And not all alleles display a dominant recessive relationship to one another. Sometimes there's this case of codominance, where both want to show itself up, or there's a case of incomplete dominance, where a mixture hybrid trait comes out when the two of them are present in a heterozygote. And lastly, of course, some alleles may not assort independently. This is a H2 concept only, because these genes may not be on separate chromosomes. We will see it later. Okay? So let's go to codominance. It's a Sec 4 concept, but I want to bring in the word incomplete dominance in case your schools did not cover it. Okay? In this case of codominance, right? In your heterozygote, typically for a big A, small a heterozygote, he will show the dominant phenotype if big A is completely dominant over small a. But when you come to codominance, in a heterozygote like big A, small a, you could have a case where the heterozygote shows the characteristics for both alleles. Or it shows the intermediate traits between the individuals with the homozygous dominant and homozygous recessive. In this case, then, now we cannot say the alleles are in a dominant recessive relationship. Okay? So, sorry, backwards. So these are the examples, right? Like ABO blood group. Blood typing is a case of codominance. And this is a case of incomplete dominance. Okay, I'll go into it, right? Now, I just want to talk to you about genetic diagrams and how you show alleles when you know that codominance or incomplete dominance is involved. In this case, you cannot do big A, small a, because it doesn't work this way, right? So what you should do is, you put the gene for it in a capital letter, and then the alleles are indicated with superscript uppercase letters, because both of them are uppercase. Nobody is recessive to anybody. Okay, I'll show you what I mean. So this is an example of cows. If you go to nice countries with cows, you will see different colored cows, okay? Cow color, for example, this is my Cow. This is a case of codominance. You cross a pure breeding red cow. Notice C is coat color, the gene for coat color. Big R stands for the allele that gives me red. White cows, C is the gene for coat color. Big W, uppercase W, stands for white. When you cross a red and white, you get a roan cow. A roan cow is heterozygote, and he shows both white and red. Okay, how do you show it? Like this. Okay, quite clear? So it's not big A, small a, so remember, if you know it's codominance, you do not write big A, small a, you write it this way. Otherwise, genetic diagrams are straightforward. See, like this, okay? Alright? So, this case, cross, one type of gamete only, right? Cross. This should be my heterozygote, right? But because I have codominant, the phenotype is a new phenotype. It's neither red nor is it white. It's actually red plus white. Roan. Okay, we call this codominance. Easy? Okay, not difficult. Only thing you take note is you must write the gametes correctly. Sorry, you must write the alleles correctly. Otherwise, the whole answer is wrong. Okay? Let's look at this one now. So let's do your roan. Your roan cross roan. What do you expect? A typical hetero, monohybrid hetero cross hetero, should give you what kind of ratio? What? Hetero cross hetero, what kind of ratio? 3:1, right? My typical Mendelian ratios, right? Let's see if it works here, okay? So over here, how do I draw gametes? One allele into each circle, right? This is monohybrid. Cross, like that. Is this 3:1? No, it didn't follow Mendel's ratios. So we call this non-Mendelian genetics. Okay, nothing very off, it's just like this. Okay, explains to us why in the world not everything follows 3:1 ratios, that's all. Okay, so this is a simple case of codominance, right? Where when you look at the outcome of a cross between heterozygotes, you don't get the same Mendelian 3:1 ratio. It deviates from the expected Mendel's 3:1 ratio when you're crossing two heterozygotes together. Okay, in fact, you get a 1:2:1, right? Which is very similar to the Mendelian genotype ratio between two heterozygotes, right? 1:2:1. Okay? So this is very common to us. We've seen sickle cell anemia so many times. And it's a case of codominance as well, in a way. Okay? Right? And if you have the sickle cell trait, you have to express both things. Alright? You are neither normal nor are you sickle cell, you are in between. Okay? And this is how we show the allele. That's why we notice, we write it this way. HB is the gene locus, capital big A for allele for the normal thing, HBS for sickle cell. That's why, now you realize why is the gene so strange, why not big A, small a? It's because it's codominant, okay? Like that. Alright? So far, alright? Now let's look at this one. There's this other strange thing called incomplete dominance. Okay, it's just splitting it a bit more finely. Sometimes, when you cross a hetero with a hetero, right, you get a nice case where you get a roan color, which is a clear-cut trait of red plus white. We call that codominance. But let's say, for example, snapdragons, you had red plants and white plants, you cross them together, you get pink. Okay, now, what do we call this? Right, we call this incomplete dominance. Alright, quite straightforward, otherwise the drawing is exactly the same. It's just how we call it. So if you can see clearly both are showing up, we call it co. It is a mixed blend between the two, like red plus white give you pink, we call it incomplete. Straightforward, right? Okay? Okay, so this concept covered, let's do multiple alleles. And ready, man? Really, so sad. Okay. So if Anna ready, then if you want to get prepared okay, for the next lecture, read this and sex linkage. Okay? Alright? Okay, see you next week.